Good afternoon. On behalf of Ed Steele, Chairman and CEO of EAS Consulting Group, I would like to welcome you to the third in a 5-part webinar series on GMP compliance in the Dietary Supplement Laboratory, presented by EAS Senior Director for Dietary Supplement and Tobacco Services, Dr. Tara Lynn Couch. EAS specializes in FDA regulatory matters with a prime focus on assisting domestic and foreign food, pharmaceutical, dietary supplement, medical device, cosmetic, and tobacco firms comply with applicable laws and regulations. EAS is staffed with former FDA compliance and inspection officials and industry executives, and is assisted by an extensive network of consultants with many years of FDA and industry experience. Today's webinar on what is a scientifically valid test method is presented by Dr. Tara Lynn Couch. Dr. Couch is a PhD analytical organic chemist with over 25 years of diverse laboratory and regulatory experience in academic field, contract, and manufacturing environments. She's a sought-after expert on issues pertaining to quality control in both pharmaceutical and dietary supplement manufacturing facilities, including the establishment of specifications and the development of well-organized, sophisticated laboratories. Dr. Couch joined EAS as an independent consultant in 2012, became an independent advisor for dietary supplements in 2015, and recently joined the company full-time as a senior director. Over the years, she has assisted numerous dietary supplement companies with the development, improvement, and implementation of strong quality systems that are scientifically sound, efficient, practical, and compliant with all FDA regulations, and has performed mock FDA inspections, gap analysis, and contractor facility audits. As a reminder, during the webinar, you may ask any questions for Tara by typing them into the chat box. With that, I will say thank you again for joining EAS, and I'll turn the presentation over to Tara.
All right, good morning or good afternoon, everyone. So this is Dr. Couch, and today we're going to have the third part in our series, as Victoria mentioned, on laboratory operations, and we're going to discuss what a scientifically valid test method is. So what we're going to be talking about is what's happening in the quality control laboratory. So this can be an internal laboratory or an external contract laboratory that's being used. These laboratories provide analytical and or microbiological testing for dietary supplements and their ingredients therein, also in process samples, and they may provide other services such as method development and verification, which we're going to talk about today, as it pertains to analytical and microbiological laboratories. Some laboratories also will hold storage of stability samples and conduct that testing for you. So in 21 CFR 111, which are the current good manufacturing practices in manufacturing, packaging, labeling, or holding operations for dietary supplements, Subpart E, which discusses testing, 75D1 and 75H1 both reference what it refers to as scientifically valid test methods. Scientifically valid methods are also discussed in Subpart J of the regulation, which is laboratory operations. Despite these references to a scientifically valid method, the FDA does not define what a scientifically valid method is in Subpart A, which is the general provisions or the definitions section of the regulation. However, in the preamble to the regulation, if you've never reviewed the preamble, it's an 800+ page document that addresses all the questions that the FDA received during the comment period, and here's a link to that, that preamble. And it does describe what a scientifically valid test method is in the preamble. On page 204, it states that a scientifically valid method is based on scientific data or other published results, such as those that would be found in scientific journals, textbooks, references, or some other sort of proprietary research. And then it goes on to say on page 205 that compendia can serve as a role in establishing test methods, but just because they're compendia does not mean that they can solely be used, because the scientifically valid method has been described on page 401 as having 4 attributes: accuracy, precision, specificity, and consistently doing what it's intended to do, which we refer to as ruggedness. So some compendial sources that can be utilized for dietary supplement methods are the USP, of course, the United States Pharmacopoeia. So they have a large number of test methods that can be utilized. The Association of Analytical Communities, or AOAC International, also has a wide variety of test methods, both analytical and microbiological. The Bacteriological Analytical Manual, better known as the BAM, is the FDA's guidance for microbiological test methods. The Food Chemical Codex, the FCC, which is known as the Brown Book, also has a variety of test methods, specifically as it pertains to flavors and colorings and other sort of additives in the dietary supplements. And no one knows better how to analyze pesticides than the EPA. So the Environmental Protection Agency has a lot of different test methods available for that. So I would caution with all these compendial methods, and as mentioned on page 205 of the preamble, just because the method is compendial does not mean it may be applicable to the dietary supplement product or even raw material formulation, and that has to be demonstrated, and we're going to talk about that in just a minute. And because these methods are compendial, they sometimes tend to be a little bit archaic because they do go through a scrutinized process. AOAC methods, for example, have to go through studies with 7 different laboratories to verify that the test methods are appropriate. So sometimes they're not the most current type of technology and don't use the most current type of solutions and that sort of thing. Just for comparison purposes, in 21 CFR Part 211, which are current good manufacturing practices for finished pharmaceuticals, it also discusses the validation of a test method. So it talks about the accuracy, sensitivity, specificity, and reproducibility in 21-165. It also talks about stability testing in 166, and it talks about having data that's accurate and reliable and suitable for its intended use in 194. Pharmaceutical products and their corresponding test methods must be pre-approved by the FDA, and this applies to new drug applications and abbreviated new drug applications, or NDAs and ANDAs. This is not the case for dietary supplements, so it behooves us to have that data to demonstrate that. And those compendial methods apply for pharmaceuticals because the USP is law to follow those regulations that are there. For dietary supplements, following the USP is completely voluntary. So there is the Dietary Supplement Compendium, better known as the Green Book, and that has a variety of test methods in there, but those are not required to be followed by law unless you're using going through the USP verification program, and then that would be a requirement. So there are a variety of different guidance documents that talk about how to validate a test method. So here are just a few of them I've listed for your reference. So CDER, the Center for Drug Evaluation and Research, has several of them. So does the Center for Veterinary Medicine and the Office of Regulatory Affairs. And even the USP Chapter 1225 has some recommendations for validation of compendial methods. There are also other guidances from the FDA, from different offices. There's a guidance for industry from the ICH, which is the Q2B, Validation of Analytical Methods, that came out quite some time ago in 1996, but is still a good method. If you're talking about chromatographic methods, there's also some references specific to those. So there are a variety of different guidelines that can be used. So typically what all of these will cover in these particular guidance documents, all pertaining to pharma, and we're going to contrast those with dietary supplements in just a moment. It covers these criteria: accuracy, precision, specificity, linearity, quantitation limit, detection limit, ruggedness, robustness, forced degradation experiments, standard stability, and sample stability. So these are all listed here. So this is what would be required on the left column. These are all the different methods that would be discussed in those guidance documents that pertain to pharmaceuticals. Now, if we take what comes out of the preamble, we just have 4 of those categories of the, 12 that are listed here on the left. So we're going to focus our attention on these 4 that sometimes are referred to as a method verification as opposed to a method validation. It's just a nomenclature. It doesn't really make any difference as long as you state which criteria are being assessed. So accuracy is the closeness of a test result obtained to the true value. So this is usually demonstrated by fortification or spiked sample studies. So what one does is fortify placebo of the product matrix that you have, or even raw material matrix that you have, with a known concentration of an analyte on top of that. And then you analyze the sample and determine how much of that is recovered. So it would be determined as a % recovery. This is typically done at 3 different levels. You'll often see 75%, 100%, and 125% of the expected recovery of the analyte. However, for dietary supplements, we frequently have to have significant overages to account for meeting 100% of the label claim, and that's testing to 100% of the label claim, because dietary supplements are a category of foods. So we often would have to extend those ranges pretty significantly. So for a gummy situation, for example, those overages are typically 200 to 300%. So we would have to cover a much, much larger range in order to accommodate for those types of analyses. Precision is another requirement that we would use from the preamble, and so this is the degree of agreement among the various test results. So you have a flashback to when you had Gen Chem or some sort of statistical class. So remember doing the standard deviation. So that is the equation for standard deviation, the S equation. Now we can luckily just do this in Excel or do this on your calculator very simply without having to plug all it in. And what you do is you compare that to the mean, and that gives you a % relative standard deviation or a % RSD. And this is typically what you would evaluate for your precision criteria. So in order to have any true statistics, you have to have at least 3 samples that are analyzed. But of course, the more replicates that you do, the more accurate your results are going to be. So some people will use the rule of 5, some people will use a rule of 10, some people will use a rule of 7 that comes from the Barr decision. That really doesn't matter as long as you have at least 3 and you define what those criteria are. Specificity is the ability to be able to assess an analyte in the presence of components that are expected to be there, so i.e. the product formulation or the raw material component. So what you would do is prepare this placebo without the analyte present and determine if you have any interferences. So there are a variety of different interferences that can be present. So just a complex matrix can hide the analyte of interest. So this is often the case if you've got a multivitamin, multimineral, multibotanical, what I would like to refer to as the kitchen sink type of product. There can also be ingredient-ingredient interactions, such as oxidation, that can result in additional analytes or loss of the analyte. And those could can also hide the analyte. There can also be instrumental interferences that may be present. So all these things need to be taken into account, and that's why we do specificity experiments. Ruggedness, or second stage reproducibility, it's sometimes called. Reproducibility is obtained with the same sample on a variety of environmental conditions. So that's typically different analysts analyzing the sample on different days, using a different piece of equipment, using different standards that are prepared. And basically what you would do is take this into two parts and take a sample that's been prepared and send it 2 different directions and have it analyzed. And then you compare the % difference from one analyst to another analyst. So this is that consistently does what it's intended to do. So that's what we're driving at, to try to figure out how consistent is this method going to be over time and in real world applications. So again, going back to the comparison of pharmaceutical type validation versus dietary supplement quote verification, we've covered these 4 categories of accuracy, precision, specificity, and ruggedness. But again, there were a variety of other ones that could be addressed, of other topics that could also be addressed, and some of these are very important to do in a dietary supplement verification experiment. One of those is linearity. So the linearity is the ability to obtain an analyte response that is directly proportionate to the concentration of an analyte. So this can be very beneficial, especially in dietary supplement laboratories, but because we often have different types of raw materials, different types of products, different types of in-process samples, different types of testing that we're doing that covers a wide range of concentration of the analyte. And it's usually desirable to try to analyze all those at the same time in one set, if at all possible. So that's where linearity would be important to demonstrate that. So linearity is usually also added as a criteria for that scientific verification experiments. So what you do then is you basically shoot various concentrations of those analytes and determine a linear fit through those using a correlation coefficient, and you back calculate out the concentration to make sure that it fits back onto that line. So the use of multiple samples is multiple analyses is usually preferred. So the FDA really likes the rule of 3, which comes from pharmaceutical. So if you run 3 different calibration curves, that usually serves the purpose of demonstrating linearity over the range that you want to use it at. Another factor that can be very important, especially when we're talking about trace analysis or the contaminant section that we have to cover for dietary supplements. So this could be your pesticide analysis, this could be your gluten analysis, this could be heavy metals, residual solvents, all of these types of things where we're looking for a small amount of something. So in that case, the quantitation limit of the method is very important. So the quantitation limit, the QL or the LOQ, depending on your background, is the lowest amount of an analyte that's in a sample that can be quantified with acceptable accuracy and precision. So you prepare a known concentration of the contaminant, and you analyze it with your test method and determine what that quantitation limit is. So typically you'll start at a signal to noise ratio of approximately 10, because that's typically where the quantitation limit is defined theoretically. And then you can lower that or increase that depending on what kind of response you're getting on the particular piece of equipment. And again, we're going for a specific level of accuracy and precision with those. So 10 isn't necessarily what we're going for. We're going for accuracy and precision, but that gives you a ballpark of where to start. Standard and sample stability can also be really important. So to evaluate the stability of a prepared standard, so you know how long you can use a standard after you've prepared it, or if it's something that has to be prepared every time you go to prep a set of samples. So in a good scenario, this can take 2 to 8 hours depending on the analyst and the complexity of the samples, of the standards. But in some cases, for example, if you're running a pesticide screen of something like 100 different pesticides, it can take you weeks to prepare a standard with all those different analytes present in it. So you certainly don't want to have to make that immediately after you make the other one. You would constantly be making those. So you want to do some sort of stability and verify that that standard can be utilized for a specific amount of time. It also applies to samples. So we were talking about standards in that particular case, but for samples, the same thing is kind of true. So let's say, for example, you load up all your samples to run over the weekend, which if you're running smartly in your laboratory, that's something that you're certainly going to do, or running overnight. What happens if you run out of power or the instrument shuts down or something happens like that? Can you re-inject those samples, or do you have to re-extract those all over again? So what is the situation with that? Once the vial has been punctured, if you're running a chromatographic analysis, is that something you can just start up and do again? So that's a question that you need addressed. So for efficiency in the laboratory, sample stability studies are very commonly done because it really impacts the way that the laboratory operates. So what you do for either standard or sample stability is you take 2 portions of a single standard that's been prepared and you run it in different directions. You can store it in ambient conditions, like out on the counter or the laboratory bench. You can put them in the refrigerator. This can be a separate unit, or this could be a refrigerated auto sampler, or you can just leave it in the auto sampler. Then what you'll do is you'll reanalyze those samples that have been sitting there with a freshly prepared standard or sample and analyze those and calculate the % difference. And you just continue this until you no longer meet your criteria of whatever that might be, plus or minus 3%, plus or minus 5%, whatever the situation would be. Most of when we talk about doing method verifications, a lot of people think only about analytical test methods, but this is far from the case. So this is very important also for microbiological test methods. So for microbiological methods, the media has to be verified to be sterile. So this is sterility testing. So most of the time we have dehydrated media that would be hydrated, even if we have pre-purchased media that's been shipped to us. You still need to demonstrate that that media is sterile and doesn't contain any growth from the get-go. So this usually involves incubating a representative sample of each lot of the media at, depending on the test method. If it's for total plate count, for example, it's typically at 35 degrees, plus or minus 2 degrees Celsius, for up to 14 days. So some media will dry out in that length of time, and that's not appropriate. So some people will only go for 7 days, but this would be something that would have to be determined by your microbiologist. But you want to make sure that your media is sterile. So this is sterility testing. On the flip side of that, you want to make sure that if there was bacteria, yeast and mold, or some sort of fungi, if that was present, you'd want to make sure that it is in fact going to grow, so that that media serves as appropriate food for that particular organism. So growth promotion studies are done. So this is basically putting known bacteria or yeast and mold fungi onto the bacteria and make sure it will support that growth over a specific period of time. So you inoculate the sample with that media, put it on the culture, and you let it grow for the appropriate amount of time and make sure you can recover it. The other thing to do is preparatory method testing. So we've been just talking about the media itself without your sample being present. So just like we would do fortification or spike samples on analytical test methods, you want to do the same thing on microbiological test methods. So you would inoculate the sample, your sample now, with this known bacteria and see if you can recover those. So, and often what you will find is that you will need to make modifications to the test method. So these can be test methods coming out of USP Chapter 61 and 62, which are the pharmaceutical sections, or 2023 and 2022 are the Dietary Supplement Compendium. It also can be methods coming out of the BAM. So wherever you're getting these, you may have to tweak some of these things about the method. And so what this can be is tweaking the dilution factor. You might have to modify incubation times. So this could be because your media is drying up. Dilution factors can be changed because if you have a colorant, for example, or something that has a strong color, like some of the vitamin A's or even B12, if you're using raw material B12, it's pink. So if the concentration is too high, you may not be able to see anything in that pink color. So you may have to change the dilution factor and increase that. You might even have to change incubation times or temperatures. So you want to make sure that you have sufficient growth of the microorganism, and so this is why we would do these preparatory test method studies. Sometimes there won't be any growth because some of the materials and the formulations that we make are antimicrobial. If that's the case, then this would be demonstrated doing these preparatory test methods. You may not even have to do microtesting at all, but you have to have data to support that, and this would be a means of coming up with that data. So in order to do these method verification studies, you will first develop a protocol. So this is basically a detailed written plan issued before you start the method verification that defines all the different experiments that you're going to do. So all those different criteria that we've assessed that we're going to do, those 4, accuracy, precision, specificity, and ruggedness, and any other ones that we want to add, such as linearity or quantitation limit, for example. This is a document that's prepared, that is a control document that's going to go through your change control system. So this would be approved by somebody in the quality unit. Now the laboratory is defined as a quality unit, so this would just be a second person that would be reviewing this document, and it'd be assigned some sort of unique protocol identification number. Now there may be some situations where a protocol isn't necessarily needed. One example of this is if you've been running test methods on a particular sample for a long time and you've seen recovery of micro, or you've seen recovery of analytes and done a variety of these studies, you may already be sitting on a lot of data. So you might just need to summarize that data, and so you wouldn't necessarily have to write a protocol to that, and we'll talk about that in a little bit. So what would be provided in a protocol would be the purpose. So we're going to demonstrate that this method is scientifically valid. That's what we're charged to do in the regulation. So we would have the name of this method and the designation. So this would be all test methods have to be assigned a SOP number or written procedure. So we would have that designation. What the description of the method is, this is typically what you're going to turn into an SOP later on. All materials and or products that you're going to examine underneath this protocol, and the verification requirements. So again, our 4 categories for sure, and any other ones that we might add, and microbiological growth parameters if those are required as well. And then a detailed description of your experiments. And of course, what's very critical is having the verification criteria. So say we're going to obtain a plus or minus 5% accuracy on this particular test method. So this is the goal of this method validation or verification experiment. So you want to make sure that you've got those criteria provided. Upon conclusion of executing all of those experiments, which of course would be documented in accordance to your documentation practices, and follow good documentation practices to make sure that we have integrity of our data, then we would provide a summary report. So this is just going to detail all those experiments that we ran and the results of those experiments. So just like the protocol, this eventually will be signed off by someone else in the quality unit. And again, this is a controlled document, so it would be assigned some sort of unique protocol number with some sort of revision control assigned to it. Because there may be cause to come back and repeat this, or repeat it on different types of materials or different types of products. So verification summary reports should also be prepared for existing test methods. So this is what I was referring to earlier. So if we have historical data that we can just pull all that together and dig through our records and find that and show that our method is accurate, precise, specific, and rugged, we can just compile that data and not necessarily have to conduct any new experiments. But we must have data to demonstrate that our method is scientifically valid if it's going to be used for dietary supplement release. So this applies to analytical as well as microbiological test methods. So in this summary report, you're going to have a purpose of the verification. So a lot of this is going to be regurgitated basically from your protocol, the reference to the method verification protocol and the number that was associated with it, the method name and designation, just like we saw in the protocol, what materials and products were examined, and then a detailed description of all the experiments. So we now have conducted all these experiments. Some of your analysts did a lot of work to demonstrate that these methods were scientifically valid. So you want to put all that information in there. So what apparatus was used, what equipment was used, reagents, standard preparations, placebo preparations, and all your experimental results. So tabulating these in some sort of tabulated form is also an easy way to represent that information. Provide example calculations so there's no ambiguity about what those were, how those were determined. And your statistical evaluation, so percent recovery is what we're looking for accuracy. Percent RSD, or relative standard deviation, is what we're looking for precision. And percent difference is what we're looking for in the ruggedness experiments and potentially also in some of the other experiments that you may be adding. A summary in reference to the protocol amendments or deviations. If you have to amend or deviate from that procedure, remember this is a controlled process, so you should have a system that will handle these things and say how this impacted the initial protocol. And then, of course, a statement about whether the acceptance criteria that were in the protocol were met. You'd be shocked at how many of these method validation experiments I've seen and summary reports I've seen that does not have this. The whole point was to say, is this a scientifically valid test method, and what are my criteria of this particular method? So you want to make sure that you see those in there. And then, of course, any conclusions about standard and or sample stability. These samples are good for a week. The standards are good for 2 weeks. You know, whatever the situation would be, that information would also be provided. This is also a place where we often determine what the critical factors of a method are. So if it's really vital that the pH of a mobile phase be 6.5. If that's really, really important, then this would probably come to light during these experiments, and that's something that you would want to roll into your written procedure and make sure that this is covered, and analysts that are running this method on a day-to-day basis know that this is a critical factor. And then a statement about the overall method variability, which is usually dictated from your accuracy experiments, would also be provided. In subpart J, 21 CFR 111.303, it talks about that we have to establish and follow written procedures for laboratory operations. I just alluded to that this will be the SOP that we have to write for test methods. So even if a method is a compendial method coming out of the BAM or the USP, we still have to have a written procedure in the laboratory. Nobody in their right mind is actually going to look at the USP and follow those as they are there. There's not enough detail provided in those, and it doesn't say anything about where you're going to document all of that information and the calculations that you're going to do. And so you need to make sure that you have written procedures for all of these. So these test method SOPs often run separate. If you're in a manufacturing facility, there'll be a set of just sort of general laboratory operations, but test methods usually have a sort of their own set of written procedures. It doesn't matter as long as they're written procedures and comply with 111.303. So as with any other control document in a GMP environment, it has to be approved by the quality unit or a secondary person in the laboratory, has to have a unique identification number with revision control that would run through change control if we were to make a change to this particular method. And then anyone that's going to be using this test method has to be appropriately trained on this method. So anyone that's using this will have a training record that documents they've been trained on this particular SOP. And if the equipment and instrumentation is very complex that are going to be used for this procedure, they would also have some sort of training on that sort of equipment and or instrumentation that's necessary. So every test method should have safety concerns as applicable. In the laboratory, both in the analytical and in the microbiological laboratory, there are hazards and biohazards that are present, so there are safety concerns that have to be considered. There'll be the preparations of reagents, there'll be the and/or media, there'll be the preparations of standards, there'll be preparation and the extraction criteria for both microbiological and analytical techniques. So again, critical factors would be identified here as well. The instruments that are going to be used and what those parameters for those instrumentations are, and the calculations that are going to be done, and always you should have an example calculation. So just seeing an equation is one thing, but then seeing one being walked through in the SOP is another. So what the requirements are every time they run a method. So if we're talking about a chromatographic method, oftentimes there'll be a system suitability requirement, which is basically a reshoot of the samples in a certain number of replicates to make sure that we have a % RSD, or basically have our precision before we start this analysis and waste our time. Also linearity for calibration curves, so what we would expect that correlation coefficient to be. Put example calibration curves in your SOP, so then the analyst knows exactly what to expect for the slope and the intercept and what those calibration curves are going to look like. Check standards are also something that are typically involved in analytical test methods. So this is basically a new preparation of a standard to verify the initial calculation of the standard. In a laboratory, everything goes back to the standards. So if the standards were not appropriately prepared or appropriately identified in the beginning, all of the work that we do after is for naught. Drift standards can be very important with chromatographic methods, as well as some spectroscopic methods such as ICP. So what you basically will do is take a standard and reshoot it periodically or reintroduce it into the sample and make sure that over time we're not drifting out of the controls of our instruments. This can be really important because sometimes, as I mentioned earlier, we can load up our samples on Friday night and come back on Monday and we have a pile of data, and that's perfect. That's exactly what we want. But it's been running for a really long time, so we have to make sure from the time we left on Friday night to the time we come in on Monday morning that the instrument hasn't drifted and all of our analysis are appropriate. Fortification of samples may also be something that you want to do on a routine basis. This, of course, would be part of your accuracy experiment as part of the method verification, so this may not be needed each time you do an analysis, but there are some situations in which you might want to do that. Blank injections, if you have a very large, complex dietary supplement formulation, sometimes you want to run a blank sample in between and make sure you've had an opportunity to remove all of that from the column before you proceed into the next analysis. For microbiological test media, method criteria, negative controls would be non-inoculated media. So this is different from sterility testing. Remember, sterility testing is where we incubate it for a specific amount of time, make sure we don't have any growth. So running a negative control is just a media that hasn't been inoculated with your sample, and you run it with every set. And oftentimes you run more than one with every set. So what this does is demonstrate that at the time of the analysis, there was no growth on that media, and there hasn't been any cross-contamination in the micro lab. So this is a critical issue in the micro lab to make sure that there's not been cross-contamination. So if you have a very large counter where all these samples are being prepared, and you're preparing, you want to prepare as many as you can at one time to be efficient in the laboratory. So if you've got 50 samples, maybe you want to have 5 negative controls located throughout the sample counter where you're preparing them. On the flip side of that, you may want to, you also want to have positive controls that are periodically put into your sample preparations. Now, positive controls aren't usually done with every single analysis, but they need to be done periodically to demonstrate that, yes, if E. coli was there, I'm going to see it and I'm going to recover it. And in addition, I obviously had one sample that had E. coli in it if I had a positive control, and make sure that I didn't have any cross-contamination, and every single one of my samples came back with E. coli. So that's unlikely to be the case in a sample preparation, so that indicates that something happened in the laboratory and you had a cross-contamination issue. So the FDA is well aware of the issues that go on in the laboratory, and we're getting more and more feedback from the FDA in warning letter and 43 observations. So I've got a section here to show some of those, but before I even talk about those, in having a scientifically valid test method and having that as one of your specifications, and then having testing that is done to demonstrate that, has far and away been the biggest issue of the dietary supplement for the last 6 years, or now we're almost at 7 years, since the regulations were applicable to all size companies in 2010. So over 55% of all the warning letter observations and 43 observations are related to specifications and testing. So this is a critical area in the laboratory, but also in the dietary supplement manufacturing and industry in general. So here's just a few that I found recently. So there is no evidence to show that USP microbiological test methods 61 and 62, that's those chapters for micro, are suitable for use in testing your raw materials and finished product. Remember, in the preamble, it said that just because it's a compendial method does not mean it's applicable to your product. You still have to demonstrate that it's scientifically valid. So that's that accuracy, precision, specificity, and ruggedness that we've been talking about, right? So you have to have data to demonstrate that, even if it's a USP method. The second one, you did not provide sufficient documentation to demonstrate that the test method you developed for your MSM product is a scientifically valid method. For example, they're using FTIR to assess the purity and strength of MSM. So IR is certainly not an appropriate method to identify the strength and purity of MSM. It might identify it, but it's certainly not going to demonstrate purity and strength of that. You have not demonstrated that the method used to identify probiotic species is scientifically valid. So the probiotic industry is huge right now. So yeah, you can't hardly go a couple of days without seeing a commercial for a probiotic, it seems. We note that the method in use is limited in its capacity to adequately identify live strains of organisms. So I'm not sure what test method they were using, but it obviously wasn't scientifically valid. So my guess is this was probably some sort of FTIR technique, or even some sort of microbiological technique that wasn't identifying DNA. Your standard reference material is inappropriate because it is a previous lot of product for which identity has not been confirmed. A previous lot is not an acceptable standard unless it is adequately confirmed to be acceptable as a reference standard using scientifically valid methods. Okay, remember I said everything comes back to the standards when it comes to the laboratory. So if we don't have standards that have been appropriately identified and used, then all of our data that we generate has no integrity. And then you conduct organoleptic testing to verify the identity of your powdered dietary ingredients, including butcher's broom and Lactobacillus acidophilus. The powdered nature of these ingredients, it decreases the reliability of organoleptic analysis and may lead to misidentification when it is the only test used. Therefore, organoleptic analysis is not by itself an appropriately scientifically valid test method to verify the identity of your powdered dietary ingredients. That is certainly the case for any sort of botanical or any sort of probiotic species. And even if you have low concentration, you have to make sure that you're using the appropriate thing to do an analysis. So just looking at the color and the appearance and maybe the taste of something is certainly not going to identify Lactobacillus acidophilus. All right, thank you for attention, and we have a few minutes. If anyone has any questions, I'd be happy to entertain those.
Hi, Tara. Yes, we do have a few questions here. The first one is, how would you verify the microscopical testing?
Yeah, we had a question about that before. So certainly there would be, I know there are different standard magnifications that a lot of the manufacturers will supply. So that would be a means probably where I would do that with a scope and make sure you can see certain striations and such when you're using a scope, and making sure that you have the accuracy and the precision that you need to do those sort of things.
Okay, thanks. The next question was about getting copies of the slides, which I believe Amy Scanlon will get out to you probably later this week. I'll touch base with her and make sure that happens.
Yes.
I'm sorry?
I said yes. Sorry.
Excellent. The next one is several parts. It says, I have a couple questions about the identity testing of incoming raw materials. Is it possible to use FTIR for testing of enzymes, vitamins, and minerals? Do you want me to ask them at the same time? Sorry.
No, that's okay. Enzyme, vitamins, and minerals. Okay, so let's take the easy one first. So for minerals, no, because the way that the FTIR works is it's looking for functional groups in an organic material. So it's looking for the stretching and the rotation of different types of functional groups, such as a carbonyl, for example, a carbon double bonded to an oxygen, or something of that nature. So that's what it's looking for. In a mineral, there aren't going to be any of those. So the mineral doesn't have any of those carbon bonds and stretching. So FTIR is not applicable to minerals at all. Now, if a mineral is on top of a carrier of some sort, if the carrier is organic, it could potentially identify what the carrier is, but we still have not identified the mineral. So the mineral would have to be done in a different manner. So IR alone would not be able to identify the mineral. So you may have to do a combination of those two things in order to do that. For a vitamin, it's going to depend on the concentration of the vitamin. So a lot of the vitamins that you'll receive, like I don't know, a riboflavin, for example, or B6, are going to be at very high concentration. So 98% typically. So in that case, once you have a known standard for those vitamins, which you certainly can obtain from USP or other resources, you can get those standards, and you've identified verified that standard is appropriate, then you can use IR to do that identification. Quick tests can be done in 5 minutes. It's awesome, right? So there certainly is applicability for it, and that's one of the perfect places to use IR. Now, this may not be the case in some other types of minerals. So, for example, in the dietary supplement world, we will frequently use a titration of B12, biotin, or folic acid. And that's because the RDI for those three is very low. So we're usually using them in very, very small quantities in the formulation. And so in order to get a uniform mix of those, we need to have a higher, be able to add a higher amount. So we'll typically do like a 10% titration or a 1% titration on something. So when we're at that, we're no longer of high purity for the IR, and what you're going to see is whatever that carrier happens to be. So whether that carrier is dicalcium phosphate or whether that carrier is MCC, for example, microcrystalline cellulose, if it's one of those carriers, that's what you're going to see. You're not going to see the biotin or the B12 or the folic. So in this case, that case, the IR would not be applicable for the identification, and you would have to do probably a chromatographic analysis to demonstrate that. So the same can be true for the oils, so vitamin A, vitamin D. If they're on a carrier, which often is also the case, then those would have to be done through some other sort of means. Some of the vitamins also will be protected on a beadlet, and then you're not going to be able to do an IR through the beadlet. So you cannot crush those. That's the purpose of those beadlets. So they're very highly protected. So in a mortar and pestle, it's impossible to crush those. So that's not going to work either. So you have to make sure you evaluate that. For the enzymatic, it's the same general approach. Typically, no, your enzyme activity is going to be far too low, and it's not going to be pure. It's going to be on some sort of carrier, so enzymes cannot be identified that way, and will usually have to go through some sort of other chromatographic type of analysis in order to identify them.
Okay, and the next part of the question is, are we required to purchase reference standards or sample spectrums for each ingredient, and what qualifications does a person need to interpret the spectrum?
Okay, so the answer to that is yes. So everything goes back to your standards. So you're required to have an unequivocally identified standard. So a lot of those can be purchased. USP has a long list of those. Some of those, USP does tend to be the most expensive form of getting those, and so what a lot of people will do is purchase those and use that as their primary standard, and then they'll purchase purchase them cheaper from one of the chemical suppliers, such as the Sigma, for example, and then compare that to the USP standard and use that USP standard, use that Sigma standard, because it's far cheaper to do so. But it still is qualified against that USP standard. Now, when it comes to botanical reference materials, sometimes referred to as BRMs, in those cases, sometimes those are not very readily available. So there are some suppliers, some laboratories such as Alchemist or Chromadex that do have some of those available, but some of them you can't get. So you have to make sure that you go through the process of finding one that is appropriately identified to make sure that you can use it as your standard. So there's a variety of different ways to go about that, and we could talk about that for days, about how to go about that process. But you definitely have to have those standards.
Okay, and I'm sorry, did you answer what qualifications a person needs to interpret the spectrums? I'm not sure, I may have missed that in the...
Yeah, so this would be similar to what we're talking about with the scientific validity, right? So you have to make sure that you unequivocally identify that, right? So, and characterize that. So that's usually the terminologies that use. You've appropriately characterized that. So if it depends on what the chemical is. So you need to identify the chemical entity. So depending on what it is. And if you're not sure how to do that, then you need to utilize a contract laboratory or a consultant, someone with the appropriate scientific expertise in order to tell you how to do that and to go about that process.
Okay, thanks, Tara. The next question is, we sample each lot for each ingredient we receive, but do we need to take multiple samples of each lot and make a composite to test?
Yes, you should, because it's a requirement in the regulation, and I'm not going to be able to come up with that reference off the top of my head. I believe it's in subpart F of the regulation, but it talks about making sure that all samples that are acquired are representative and statistical. So any sample that you acquire needs to meet that criteria. So there's a variety of different ways that you can do that. So for an incoming raw material, one that's frequently used is the square root of n+1, where n would be the number of containers that you receive. So let's say you received 25 different containers, then you would go through and sample 6 of those and create a composite of those 6, and that's what you would assess, would be those 6, sample that composite of those 6. Now, when you are compositing, you have to be careful that you would not dilute out any issues that you would be finding. So you should have some sort of limit on the level of compositing that can be done. So what I recommend is, not more than 10. So if you had, let's say, 100 different containers that you receive, you would have to sample 11. So you would have to do a composite of 5 and then a composite of 6, right? So, and break those up into 2 separate ones, because you could be potentially diluting out any issues that were present. And that's the whole point of doing this testing, is to make sure that they meet your criteria that's designated on your specification. So you do need to do that. ANSI charts, they're also the American National Standard Institute. They have criteria also that can be utilized. So they have some nice tables that can be used. A lot of people will use those for their raw material statistical sampling and figure that out. But you need to have whatever it is that, whatever sort of statistical assessment that you want to use, you just need to have that documented in a standard operating procedure, and then that would be executed, and you would have documents to show which samples that you took. It's also a wise idea to maintain the integrity of those original samples, so not composite the entire thing, because if you do find a problem in, say, those 6 samples that we acquired, then you could go back to those individual samples and look in those individual ones and see if it was in just one of those samples or if it was in all 6 of them. So, and this is particularly important in micro, because certainly micro does not is not uniformly distributed in microbiological contamination. So you could have one container that got wet, for example, and you had mold growing in one container, and the rest of your lot is perfectly fine. And you don't want to have to throw out the entire lot just because one container was subject to some sort of contamination. So it's nice to maintain the integrity of those and come back and reassess that as your next step if you were to get into an out-of-specification situation.
Okay, thanks. We do have a question. How many FDA warning letters were actually regarding test methods? I guess your microphone had cut out when you were...
Oh, sorry.
That's okay.
So 55% of all the warning letter observations that have been issued every year, so 2010, 11, 12, 13, 14, 15, so all of those are associated with test specifications and testing. So i.e., that means they reference 21 CFR 111.70, which talks about specifications, and 111.75 that talks about testing. So you can go to the FDA website. They have tables that are in Excel that you can download, and then you can manipulate and see which particular subsection of the regulation was cited if you want to go through that process. But clearly this is an area that the FDA is paying a lot of attention to, because as an industry we're not doing a very good job in this area.
Okay, thanks. The next question is, do we need to prove that our methods are stability indicating, since we are using these methods to test stability samples?
If you're using your methods to test to stability samples, then yes, you'd have to do something for stability indicating. Now, if you're looking at like the ICH guideline, for example, which has some about the stability indicating test methods, if you're looking at that, you can't necessarily use that directly for dietary supplements if you have a complex matrix, because you're going to have all kinds of peaks if you're using a chromatographic analysis. So you're going to have a very messy background to begin with. So just looking at, all the peaks that might be greater than 1% of the area covered by your particular analyte, it's not going to work in that particular case, which is what ICH calls for. So you may have to be a little bit more creative about how you look for stability indicating, right? So, and then the other thing is, if we're talking about vitamin matrices, for example, vitamins degrade. That's what they do. That's how they work in the body. So trying to do force degradation studies to look for stability indicating methods really doesn't work either, because if you do that, you basically kill off everything and you have one enormous peak in your chromatographic run. So the way that you do that has to be a little bit different. So you would just make sure that you don't have anything, if we're talking about a chromatographic method, which is typically what you would be doing this for. If you're looking at a chromatographic method, you want to make sure that you still have resolution of your peaks. You don't have any fronting, you don't have any tailing, and you don't have new peaks that have shown up that would be potentially interfering. So it's difficult to simulate that for a dietary supplement as opposed to pharmaceutical. So it makes it a little bit more challenging. But you certainly should be doing that if you're using stability indicating methods and demonstrating that you don't have any issues that are coming up, 2 years down the road as opposed to on release.
Okay, thanks. And our last question is, using FTIR, how can you establish a standard when, for example, we use multiple vendors for one item?
Well, you still have to, it depends on, you should be using multiple vendors for one item, so in case something happened with your supply chain, you have a backup. So that's a smart thing to do, to have some sort of backup. But if you're just talking about a straight vitamin, for example, I mean, once you've characterized that particular standard, it'll apply to everything. It gets a little bit more complicated if you're talking about botanicals, right? So you may have multiple sources of a botanical. So you'll need to know what the growing conditions of that are, and you'll need to know if it's an extract. You have to have separate reference standards for an extract versus just a dried powder. So you have to make sure that you've established those things appropriately. And even if you're getting it from different vendors, you have certain criteria that any of the vendors have to meet in order for you to use that sample. So those would be how you would make sure and characterize that, and make sure you establish what that reference material would be based on those criteria that you have, not necessarily what your vendor has.
Excellent. Okay, well, that is it for the questions for today. We'd like to thank you, Tara, for providing an informative session. For those of you in the audience, this webinar was recorded, and should you wish to review it or suggest viewing to your colleagues, please visit our website. And you can reach out if you have any further questions.
Thank you.
Thank you, Tara. Bye.